Hello students. In today's video we're going to talk about meiosis, the process that's used in sexually reproducing organisms to create gametes, the cells for sexual reproduction. So let's start with talking about mitosis. So a little while ago we talked about how cells are able to divide, and that division goes by the name of mitosis. And that mitosis is used when a cell needs to repair itself. So like when you have a cut, it's mitosis that closes it. It's used for growth. You all started as much smaller than you are now, and your cells did not get bigger. You got more cells from mitosis. It's also used for development. Easiest situation is when you have a child that starts as an embryo, develops into a fetus, and all of the different parts of you are formed from one cell. And that change, those different cell types, is what we call development. But all of these things are reasons why mitosis would happen. But the one thing we never talk about with mitosis is that last thing of reproduction. If you're a asexual organism or a single-celled organism, things like amoeba, paramecia, euglena, they're going to reproduce simply by the process of mitosis. However, organisms that reproduce sexually, which is an evolutionary advantage, actually have a whole other process that's developed. Now, the thing about this process is that it comes from the process of mitosis. So it's actually an altered version of the things we already know. So real quick review. Over here, hopefully you remember we have the cell cycle, which is the life cycle that all of your cells go through. You'll note that they start in G1, and from G1 they do their cell job, they do whatever's going on, they get stuff going. There's a checkpoint here, the point of no return, and once they cross that, they enter S, where their DNA is duplicated, which is going to be a very important part of this whole process. At the end of the S phase, there's another checkpoint where the DNA is checked to make sure there's no mistakes, and then we get prep for mitosis, which is known as G2. And here you have your cell. It goes through this process of mitosis, and you end up with two cells. If you recall, this stuff, this G1, S, and G2 is all interphase, and this part right here that's mitosis starts here with prophase. So here, we start to see those chromosomes form, the nuclear membrane falls apart, then the chromosomes line up in the middle of the cell, and then the sister chromatids, which were created during the duplication in S phase, are ripped apart. And then the nucleus starts to reform, the chromosomes uncoil, and if it's an animal cell, a cleavage furrow happens. If it's a plant cell, a cell plate begins to form. We have two new cells, and then we go back to interphase. All right, I know that was really quick, but this is just a review of the stuff we talked about before. So a couple things that we need to think about when we get into this process of mitosis is that mitosis is for autosomal cells. That's a term that we keep using when it comes to chromosomes. Autosomal cells, if you recall, are all of your cells, except for two specific cells. They do not include sperm or egg. Any other cell in your body is an autosomal cell. Nerve cell, autosomal cell. Eye cell, autosomal. Stomach, autosomal. Muscle, autosomal. Your neck, autosomal. Everything in your body is autosomal except for sperm and egg. Mitosis is how your autosomal cells divide. Meiosis, which is the process that we're discussing as part of this video, is how your sex cells divide. And so, for us to possibly understand the complication that goes into that, we need to understand something very important about your cells. You should know at this point that your chromosomes come in pairs. So if you're a human being, you actually have 23 pairs, which gives you a total of 46. Now, the reason that your chromosomes come in pairs is because you get one from mom and one from dad. And so since you need them in pairs, every sexually reproducing organism has chromosomes that are in an even number. And so scientists actually look at the chromosome number in two ways. We look at it in terms of the number of chromosomes that are unique. So for instance, in a human being, there are 23 different chromosomes. And then the fact that they come in pairs means that we always have two of that unique number. So we say that 2n is the actual number of chromosomes. So let's take a look at this in terms of two specific examples. Okay? So for humans, n is equal to 23, because that's the distinct number of chromosomes that we have in terms of unique chromosomes. 2n is equal to 46, because that's the actual number of chromosomes. Because for each one of these, you have a pair, remember? Because one comes from mom and one comes from dad. So you always have two of the n chromosome number. So that's why human beings have 23 pairs, which is n, and 46 total chromosomes, which is 2n. If I were to do this for Drosophila melanogaster, which is the common fruit fly, n would be 2, and 2n would be 4. So Drosophila actually has 4 chromosomes, but it has 2 pairs. Okay? So n, let's do this. N is what we call an organism's haploid chromosome number, because that haploid comes from the word half. 2n is what we call an organism's diploid number, 2 being di. Di is a prefix that means 2. So down here, Drosophila's haploid number is 2. And its diploid number is 4. So all organisms have a haploid number, which is n, and a diploid number, which is 2n. So the reason this is so important is because meiosis separates pairs. So if a human has 2n chromosomes, which equals 46, that means that they have n pairs, which is 23. And this process of meiosis is going to create cells that only have one from each pair. So let's try to wrap our head around this. Over here, we have a pair of what we call homologous chromosomes. And you should remember from our previous work with Mendelian genetics that this prefix homo means same. So what you have here is an organism that has this allele right here, which is for height, on both of these chromosomes. And you'll notice that even though chromosome A and B are the same, they don't necessarily have exactly the same allele. So what same means is same traits, but it can be different versions. Or different alleles. And that's important to understand. So human beings have 2n chromosomes, which means every n is a homologous pair. So if chromosome 1 in a human being has the trait for eye color on it, one of your chromosome 1s has an eye color allele that you got from, say, your father, and your other chromosome 1 has an eye color allele you got from your mother. Since they both have the eye color allele, they are homologous. However, even though they both have the eye color allele, which is the same trait, they don't have to have the same version. You could get the brown eye color allele on chromosome 1 from your father, and the blue eye color allele from chromosome 1 from your mother. Okay? Homologous means same trait, but it doesn't necessarily mean the same version of the trait. Okay? And so this process of meiosis is all about separating these homologous pairs at random. So that way, every egg or every sperm will have one chromosome 1, but which one they have is random. Now, the last thing I want to talk about in this video is how those Mendelian probabilities stay the same. Because you can see right here, this A and B chromosome are stuck together. I'm sorry, this A and B allele are stuck together. And let's say that's the allele for asthma and eczema, something we talked about in class. Since they're on the same chromosome, these two traits should always be passed on together. But they're not. They are a little more frequently than other things, but it is not always together. And the reason for that is something called crossing over, which is basically what you see right here, where the A is going to take this B from the green chromosome and the B from the blue chromosome, and they're going to swap places. So you can see that right there. Crossing over just adds variety. And it actually is important because it allows Mendel's probabilities, even if you have traits that are on the same chromosome. If crossing over didn't happen, Mendel's probabilities would not work. But they do. We've actually observed them in real life. And so the reason they work is because this crossing over helps us take care of the fact that these alleles are stuck on the same chromosome because you only have a certain number. All right, so we haven't actually gotten into meiosis, but we're going to stop here, and I want you to try and keep in mind what haploid is, what diploid is, and what a homologous chromosome is. Those are the three huge takeaways from this presentation.